Okay, everyone. I think we're going to go ahead and get the last plenary session of the day started on the role of lifestyle and elevated genetic risk. And first, like to introduce my friend and colleague, Dr. Lorelei Mucci, is a professor of epidemiology at Harvard University School of Public Health and heads the cancer epidemiology and prevention track for the school. She is also the Deputy Associate Director of Population Science at the Dana-Farber Harvard Cancer Center. And her work largely focuses on prostate cancer and integrative molecular approaches in diverse patient cohorts across the cancer continuum. She's also PI of 2 large prospective cohorts, including the Health Professionals Follow-Up Study and the Ironman International Registry of Men with Advanced Prostate Cancer. And I've had the pleasure of also interacting with her as co-director of the AACR Integrative Molecular Epi Workshop. So she's joining us remotely today, and Lorelei, I think we're ready.
Fantastic. Thank you so much, Carrie, and thank you to the organizers of this really exciting conference. I'm delighted to talk with you about where I think some new developments are happening in trying to understand whether the inherited susceptibility of prostate cancer can be modified through a healthy lifestyle. So I'm going to give a brief introduction about the epidemiology of prostate cancer, talk about what's known about the strong role that family history and genetic susceptibility play in the risk of developing prostate cancer, and then finally thinking about some promising opportunities for cancer prevention, specifically in individuals who have a high genetic risk of prostate cancer. So prostate cancer is the most commonly diagnosed cancer among men in more than 100 countries, including the United States. In 2020, it was estimated that about 1.6 million individuals would be newly diagnosed with prostate cancer. This is a map of the global burden of prostate cancer incidence from IARC, and you can really see the large differences in geographic variation in the incidence of prostate cancer. This is driven by a complex variety of factors. Part of this is due to differences in diagnostic intensity. We believe also maybe differences in genetic susceptibility, but clearly based on migrant studies, there's a strong role of differences in lifestyle and diet in these incidence differences. In terms of mortality, prostate cancer is a leading cause of cancer death among men in 54 countries. In the United States. In 2022, 34,000 men are expected to die of prostate cancer, and globally, 468,000 individuals will die of prostate cancer. Again, you can see considerable differences geographically in the burden of prostate cancer mortality around the world. When we think about then, given this large burden of prostate cancer, what are the most promising opportunities to reduce the incidence of prostate cancer? These are the factors that I think most epidemiologists would agree on, so that individuals who are older age, are at greater risk of developing prostate cancer. Individuals of African ancestry, Black men have about a 2.1-fold greater risk actually of dying of prostate cancer, of developing prostate cancer, and a 1.6-fold greater risk of dying of the disease compared to white men. Individuals with a strong family history, not only of prostate cancer, but also we're starting to learn of family history of breast cancer, also are at greater risk of prostate cancer overall. And we've now, and I'll talk a little bit about what we know about how much of that family history is due to inherited genetic factors. We also think that taller height is a strong risk factor for the incidence of prostate cancer. One of the take-homes from this, however, is that what you can see is that in terms of the incidence of prostate cancer overall, none of the established risk factors appear to be modifiable. So what do we know about family history and heritability of prostate cancer? Well, there early on from data back from the 1980s, there's been evidence of a role of family history of prostate cancer in an individual's future risk. And this is data from Sweden looking at how the risk of prostate cancer in the general population, which is denoted in this dotted line, compares to those with different aspects of family history. So in this population, the lifetime risk of prostate cancer was about 18%. That's about a little bit higher, about similar though what we see in the United States. However, if an individual has a family history where a brother has prostate cancer, that lifetime risk of prostate cancer doubles. And in fact, you can see with increasing number of family members with a history of prostate cancer, you can see an increasing cumulative incidence of prostate cancer across one's lifetime. So strong role of family history in predicting future prostate cancer risk. Well, our group and others have tried to understand the extent to which family history can be explained by inherited genetic factors. And so leveraging data from a large cohort of twins from the Nordic countries, we looked at the extent to which family history can be decomposed into that part that's shared environmental factors versus those that are due to inherited genetic factors. So looking at individuals with of male twin pairs, both identical twins, which share, in theory, 100% of the inherited genome, and dizygotic twins or fraternal twins that are as similar genetically as other siblings. What you can see here on the left panel is what's the cumulative incidence and familial risk of prostate cancer. So in this cohort, the cumulative incidence was 10.5% in the whole cohort. But if a twin pair was in a dizygotic twin pair and their brother developed prostate cancer, that lifetime risk went up to 22%. And if they were identical twins and their twin brother had prostate cancer, their own lifetime risk was 3.8 times higher, or 38%. So that translated to a heritability of prostate cancer, which is interpreted as the variability in incidence of cancer that can be explained by inherited genetic factors, was 58%. And when we actually compare cancer heritability, both from twin studies as well as from genome-wide association studies, what you can see here is that prostate cancer is the cancer with one of the highest estimates of cancer heritability of other different cancer types. So it really is a cancer for which one wants to know, is the inherited susceptibility of prostate cancer fatalistic, or is it modifiable? And just to add, now there's been a large number of genome-wide association studies that have been done, and this is work from one of the more recent publications from David Conti, Chris Heyman, and others. We were a part of this consortium from the practical study. And what was really unique about this study was not only that it brought together large numbers of prostate cancer cases and controls, but it also brought together multi-ethnic populations in order to identify a polygenic risk score of about 269 individual genetic variants that could nicely discriminate future risk of prostate cancer, not only in European populations, but also those of African ancestry, East Asian ancestry, and Hispanic individuals as well. And what you can see here is these different colors refer to deciles of genetic risk based on the polygenic risk score. And what you can start to see is that individuals in the lowest genetic risk score, their lifetime risk of prostate cancer is less than 5%. However, for those in the top 10 decile of genetic risk, that absolute risk of prostate cancer is closer to about 30 to 40% in different populations. So again, really starting to think about the way in which genetic risk really drives the incidence of prostate cancer. Some additional work that we've done, and this was important to do because prostate cancer is really unique, both in terms of its prognosis, but its etiology and being very clinically heterogeneous, meaning that while many men get diagnosed with prostate cancer, more men will die of the with the disease than from the disease. So trying to understand genetic basis of prostate cancer in terms of more clinically significant aspects of prostate cancer, as well as fatal forms of prostate cancer, is quite important. I think this figure just really illustrates the fact that this polygenic risk score seems to equally predict high-grade, potentially fatal prostate cancers, as well as the future risk of lethal prostate cancer compared to those with a low polygenic risk score. So then the question that is, as I mentioned, that this study is looking at, or this presentation is looking at, how much can we offset that genetic susceptibility of prostate cancer through engaging in a healthy lifestyle? And so one of the earliest publications came from Stacy Loeb and colleagues looking at within the PLCO cancer screening trial. So this particular study included GWAS individuals, including 1,230 incident prostate cancer cases and 1,361 controls. This was at the time, now about 7 years ago, and there were 20 prostate cancer risk alleles that had been validated at that point. So we're at a much different point now, but this was looking at the first 20 prostate cancer risk alleles. And when you looked at genetic risk based on these 20 prostate cancer risk alleles, there was a tendency that in terms of the risk of non-advanced prostate cancer, in those men who had the highest genetic risk, those who had a high vegetable or fruit intake, as well as specifically high intake of lycopene, seemed to have a lower risk of more non-advanced forms of prostate cancer, as well as prostate cancer overall. This screening trial at the time did not have data or insufficient numbers on lethal prostate cancer to ask the question about, can diet and lifestyle modify the risk of lethal prostate cancer in those at high genetic risk, but really intriguing preliminary data. So for the current study that I'm going to present, we leveraged 2 cohort studies of men for whom we had a large number of the existing GWAS data to define a polygenic risk score, as well as long-term follow-up for prostate cancer incidence and for mortality. So the first study was the Physicians' Health Study, which originally actually began as a randomized control trial of 22,000 individuals looking at aspirin and multivitamins, and they've been followed prospectively now since 1982. There was a blood that was given at baseline by 14,900 men, and of those, there were 1,968 for whom we had available existing GWAS data, of whom there were 856 individuals with prostate cancer, including 184 lethal cancers. And there has been questionnaire data about diet and lifestyle collected prospectively in the cohort. For the second cohort, we looked at the health professionals follow-up study. So this is the Physicians' Health Study actually enrolled physicians into the study. The health professionals follow-up study enrolled other types of health professionals, veterinarians, dentists, optometrists. It started with 51,000 cancer-free men at enrollment. And then there were 2 different time points where DNA were collected. The first in where a blood sample was given between 93 and 94. And then there was a buccal cell collection that was done around 2004. To date, we had data available for 10,443 individuals with existing GWAS data. And of these, 2,152 were prostate cancer cases, including 251 lethal cancers. Again, updated lifestyle and diet data along this course that we could look at defining the high incidence of prostate cancer based on both genetic factors and lifestyle. So we used that polygenic risk score that I mentioned earlier that was developed in multi-ethnic populations, 269 individual genetic variants, to look at defining genetic risk in individuals with a high, moderate, or low risk. And then in terms of healthy lifestyle factors, we decided to use an existing healthy lifestyle score that was developed by Stacy Canfield and colleagues. that looked at factors specifically for the prevention of lethal prostate cancer. So the score was simple. It was a score of 1 versus 0, based on 1 if you had, for example, a healthy body weight, 0 if not, 1 if you were either a never or distant former smoker, 0 if not, 1 for those vigorously exercising. And then we had 3 components of a healthy diet based on low red meat intake, fatty fish intake, and lycopene intake. And so an individual could have a score ranging from 0 to 6 based on this 6 components. So this is a distribution of what the healthy lifestyle score looks like in our population. About 29% of the individuals in our analysis had 4 to 6 of the most healthy components of this healthy lifestyle. 41% had 3 and were considered moderately healthy, and then 30% had 0 to 2 less healthy components. And what you can see in this table below looks at what are the significant contributors to healthy lifestyle. So compared to other cohorts, our cohort has a relatively lower prevalence of obesity based on body mass index of 30 kilograms meter squared. In those who are the most healthy, we had 100% had a BMI less than 30 compared to 86% of those who are less healthy. But you can see pretty big differences in smoking status. So overall, less than 3% of our cohort were currently smoking. So this is really reflecting mostly individuals who had quit smoking in the last 10 years. And so in those healthiest lifestyle, there were about 95% either were never smokers or had quit more distantly, compared to 30% in the less healthy. And then you can also see comparisons for vigorous physical activity, tomato consumption, processed meat intake, and high consumption of fatty fish. So this table here looks at the relationship of just the healthy lifestyle score on its own compared to lethal prostate cancer, comparing those in the least healthy, because they had a healthy life score of 0 to 1, compared to those with increasing healthy lifestyle scores. And I present it both for the health professionals follow-up study as well as the physician's health study. So first, what you can see is with increasing adherence to that healthy lifestyle score, there was a reduction in the risk of lethal prostate cancer in both groups. So those with the healthiest lifestyle component had about a 50 to 60% lower risk of lethal prostate cancer compared to those of the least healthy. So next we wanted to know, well, what about those individuals based on genetic susceptibility? So this figure here compares the panel on the left are individuals in the bottom 25th percentile of the polygenic risk score, or those at low genetic risk, compared to the right panel are those at high genetic risk. And first of all, I think what you can see is that just comparing the two figures, that overall the cumulative incidence of prostate cancer overall is just really substantially elevated in those at high genetic risk versus low genetic risk. But however, what you can see is that at least for overall prostate cancer, we did not see that the healthy lifestyle benefited individuals in either group. And that's actually not surprising. When we look, as I mentioned, in terms of what we know about overall prostate cancer, we don't really see much in terms of modifiable factors. However, what we did see was when we studied specifically lethal prostate cancer, if you look at the panel on the right-hand side, you can see individuals who had the least healthy lifestyle had a lifetime risk of 6.1% of lethal disease. In comparison, the lifetime risk of lethal prostate cancer in those with the healthiest lifestyle was closer to 2.2%. And that translated to a hazard ratio of 0.55, or 45% lower risk of lethal prostate cancer. We also saw suggestions of a benefit of the healthy lifestyle in individuals who had the more moderate genetic risk, so 50 to 75%. However, interestingly, we did not see a benefit of individuals in low genetic risk, and that may be because the risk of prostate cancer overall is so low. So that risk of, there's not much additional room for a benefit of healthy lifestyle for lethal disease in those with the lowest genetic risk. The other, when we looked at the individual components of the healthy lifestyle, the two factors that seemed to contribute the most in terms of benefit were those who maintained a healthy body weight as those who were most physically active. Oh, sorry. And so finally, that work was led by Anna Plim, who was a postdoctoral fellow in our group, who now actually has just started her own independent faculty position at the Karolinska Institute. She's now validating this work within the Malmo diet and cancer study among 10,000 Scandinavian men. And what is really interesting and promising to see was that these results seem to recapitulate. So the table on the left looks at the first few rows are looking at the association of the healthy lifestyle score in individuals at low genetic risk as well as those at high genetic risk. So in both groups, seeing a benefit and reduction of lethal prostate cancer in those with a healthy lifestyle. And then the figure on the right looks at the cumulative incidence, comparing those, cross-classifying both on genetic risk and the lifestyle score. So you can see the light blue curve are those who are in the low genetic risk group and have the healthiest lifestyle behavior. Dark blue are those low genetic risk, but more unhealthy. And you can sort of see this increasing cumulative incidence. So I think one thing that's a little different from our study than this study was here it seemed to see that there was actually a benefit of the healthy lifestyle score, even among those with the lowest genetic risk. What we're wondering, and again, these are preliminary data, so we ask, please don't quote these data, is that a larger component of the healthy lifestyle in this population was smoking status compared to some of the other factors. So that may be explaining this additional benefit, but really, really interesting. So just to kind of wrap up, thinking about summary and future directions, prostate cancer does have the highest estimate of heritability of any cancer. And I think what we've shown is that these common genetic variants can be useful to risk stratify future prostate cancer risk and lethal disease within multi-ethnic populations. However, I think our data, both in the health professionals follow-up study, the physicians' health study, the Malmo cohort, as well as that early study from the PLCO, suggests that genetic susceptibility may not be fatalistic. And in fact, there may still be potential prevention through healthy lifestyle. So this idea that there may be opportunities for precision interventions and prevention in individuals at high genetic risk. And I think this is particularly important given the fact that family members of individuals with prostate cancer in their families often will go to their doctors and say, what can I do to offset my own potential future risk of prostate cancer? So I think that's something really important. I think there's a lot of opportunities to look at other things, for example, medications such as statins and aspirin. And then we're in the process of looking at not only individual dietary factors, but looking more at dietary patterns such as the Mediterranean diet, the hyperinsulinemic diet, and other dietary patterns. I think one additional component is the importance of investigating these types of questions within multi-ethnic populations, particularly so since we know, unfortunately, given the racial disparities that exist in prostate cancer, the prevalence of this polygenic risk score is much higher in individuals of African ancestry. So with that, I'd like to really thank an incredible team of people that have been involved in this work. And as I mentioned, particularly Anna Plim, who's leading this effort, including with funding not only from the National Cancer Institute, our SPORE in prostate cancer, but as well from the Prostate Cancer Foundation. So thank you so much, and delighted to take questions later on.
Recording in progress.
Okay. Do you want to do? Yeah. Lorelei, do you mind if we take a question or two right now?
I would love that. Yeah, that
Okay.
would be fantastic.
Okay. Go ahead, Kristy.
Hi, Lorelei. This is Kristy Brown from Weill Cornell. I really enjoyed your presentation, and I was wondering if you could comment more on the various genes that were studied in this context. Do you have specifics about which ones might be more modifiable than others?
Yeah, no, that's a really great question. And it was something that we didn't include in the first publication because we wanted to try to delve into this more deeply and potentially in larger cohorts as well. But one of the specific areas of importance in prostate cancer as well as other cancers has been in 8q24. And when we looked specifically at the healthy lifestyle as well as the individual components, it looked like when you looked at the individual SNPs, and there's a number of SNPs within the 8q24 that seem to predispose individuals to prostate cancer risk, there seemed to be a particular benefit of the healthy lifestyle. That's also quite interesting because the prevalence of some of these genetic variants, again, if we look at African American men versus men of European ancestry, the prevalence of these individual variants in 8q24 also seem to be important. So that's one set of SNPs in a specific genetic region that seems to be potentially susceptible to this lifestyle. And again, I think that would be really interesting also to look at other cancer types, since we know 8q24 seems to be playing a role, for example, in colorectal cancer, breast cancer, and other cancer types.
Hi, Lorelei. I don't know if you can see us, but this is Neil Iyengar from MSK.
Hi, no, I can't see you, but nice to have you ask a question.
Yeah, thanks for your great presentation. With regard to the post-diagnosis setting and some of the population data that you showed, is it possible, or have you been able to parse out the treatment intensity effects? In other words, we know that the hormonal therapy induces metabolic dysfunction, and can you look at intensity of intermittent versus non-intermittent suppression and other treatment modalities that may or may not be contributing to prostate cancer death from a metabolic standpoint?
Yeah, that's a really great question. We have started to look at, independent of the polygenic risk score, we've started to look at whether something like physical activity or some of these healthy dietary patterns, specifically in individuals on androgen deprivation therapy. Because as you say, one of the big survivorship questions is, can we improve survivorship and also reduce not only lethal prostate cancer, but things like cardiovascular mortality or the incidence of cardiovascular disease. So we've been starting, again, not in the context of the polygenic risk score, but looking at potential opportunities. And I think some of this work is substantiated in smaller, clinical trials that have looked and shown benefit. I know, for example, Christina Daly-Conright has shown in men on androgen deprivation therapy that engaging in a physical activity intervention could lower some of the cardiometabolic effects of ADT. So I think there's a lot of promising there. The question's a great one, and I think it would be really important to look at, even bringing the genetics in there. Are there individuals who have a genetic susceptibility to some of the increased cardiometabolic effects of ADT? And then again, how can you offset that? I think it's a great area for growth and research, and a really important question that you raise.
Thank you.
Okay. Yes, we have time for another question.
Thank you. Hi, Sam Orange from Newcastle University. Thanks for a great presentation. A real quick question. I wonder if you can comment on the findings from the ACSM roundtable report that showed that pre-diagnosis physical activity wasn't associated with cancer mortality, but post-diagnosis physical activity was quite strongly associated with cancer mortality. I wonder if you could comment on that, whether is there a confounding due to screening and health-seeking behaviors or something like that?
Yeah, no, that's a really fantastic question. And, I think kind of related to that, when we think about any types of lifestyle or dietary factors after cancer diagnosis, is it that people who are healthier and are able to engage in something like regular physical activity, and is that where you see the benefit? So I think some of the studies to date, I think it would be really important to look at the extent to which, for the pre-diagnostic activity, and are you talking about prostate cancer specifically, or cancer mortality?
Yes, prostate cancer, yeah.
Yeah. So I think, I think part of that, I think you're right. You have this sort of interesting confounding factor where the people who are going to be engaging in the most physical activity are also most likely to get screened, and therefore, in terms of a benefit for mortality, you have to really make sure that you carefully control for that. And so I think as a field, we need to look across all those studies, see the extent to which something like PSA screening has been accounted for. But then, as I said, also need to make sure that we're carefully accounting for post-diagnostic differences in the ability to exercise. I do think the data suggest in studies that have accounted for differences in screening, there is a suggestion of a benefit of pre-diagnostic physical activity for cancer mortality, but I think it needs to be looked at more systematically.
Thank you.
Okay, we will move on to my co-chair, Kristy Brown. Kristy Brown is an associate professor of biochemistry and medicine at Weill Cornell Medicine, and she was recruited there in 2017 after a storied career in Canada and Australia. And her current work focuses on the mechanisms whereby obesity leads to the increased risk of breast cancer and how adipose tissue causes cancer cells to grow faster, to be more aggressive, and to identify ways to use these discoveries to develop effective therapeutic options for breast cancer.
Thanks, Carrie. It's a real pleasure to be here. It's actually my first AICR meeting in person, and I've really been enjoying every aspect of it. So my lab for a long time has been interested in trying to understand the molecular links between obesity and breast cancer. And the work that I'll be telling you about today is related to some of our more recent efforts, trying to understand whether obesity may also be able to impact cancer penetrance in women who have a high risk due to genetic predispositions. So it's really nice to be able to follow Dr. Mucci in this sense, because I think this is a relatively new idea when we think about individuals who have very high risk of cancer. How could anything be modifying their risk? And I think we're slowly learning that this might be the case. That being said, I do want to approach this with some humility. What we'll be talking about today is, some biological effects that we've observed, but by no means does this show definitively that there is an impact. And we're hoping to be able to gain more insights as time goes by as to whether there are biological mechanisms in place that might lead to increased cancer formation. So it'll come as no surprise to you that obesity is a risk factor for cancer, and in the general population, it's associated with an increased risk of developing hormone receptor positive breast cancer after menopause. In order to start thinking about how that might be or why that might be, many people have focused on systemic factors that are altered with obesity, as well as factors that are produced locally within the breast. And it's not surprising to think that the breast microenvironment might actually contribute to the development of cancer, given that the breast mammary glands, which are the site of origin of these breast cancers, are surrounded by and embedded within a substantial amount of adipose tissue. What we also know is that this adipose tissue of the breast undergoes substantial changes in the context of obesity, as we see in other adipose depots. So this is considered a subcutaneous adipose depot, and with obesity, we see hypertrophy of the adipocytes. This leads to an increased production of the adipokine leptin, a decreased production of adiponectin, as well as the recruitment of immune cells, including macrophages, when these adipocytes become dysfunctional and die. And as a consequence of this, we create this chronic low-grade state of inflammation within the breast adipose tissue that has been believed to drive the growth of many breast cancers. In addition to this, and one of the key aspects of hormone-dependent breast cancer, is that cells within the breast especially the preadipocytes or the mesenchymal stem cells, are responsible for expressing the aromatase enzyme. And this is particularly important in postmenopausal women when gonadal steroid production has ceased. And we know that many of the factors that I've mentioned before, including adipokines, inflammatory mediators, et cetera, will drive the expression of aromatase within the breast. And as a consequence, we're really creating this hormonal milieu that will support the growth of tumor cells. Once these tumor cells are there, they will also feed back to the microenvironment. They'll further stimulate aromatase expression, they'll stimulate lipolysis in fat cells, and certain tumors will actually rely on that release of free fatty acids for their growth. But when we talk about risk, we don't necessarily talk about tumorigenesis, because risk is really the detection of tumors. So this is due to a combination of multiple factors. So the initial stages of converting a normal epithelial cell to a tumorous epithelial cell is one of the most important aspects of this. But really, when we're talking about detecting cancers to be able to assess risk, we're also evaluating, cancers that have become detectable. So this is a function of, how much they've proliferated, but also how immune surveillance has changed over time. And it can also be affected by the detection modalities that may be influenced by factors like obesity. So a little while back, someone relatively close to me said, yes, obesity produces factors that are known to stimulate the growth of breast cancers, but there's very little evidence to say that obesity can cause cancer. So this is when we decided to pose that question: can obesity actually cause cancer? And the hypothesis here was that if obesity could cause cancer, it must be causing changes to the DNA of the normal epithelium that could lead to tumor formation and cancer growth. So to start studying this, we decided to start looking at BRCA1 and BRCA2 mutation carriers. And I have to admit, when we started this work, this came from a purely scientific perspective. We were interested in seeing whether there might be damage to the DNA in the normal epithelium. And normal cells actually will repair DNA very quickly, so it's hard to detect it, especially when it's a single event here and there. And we were hoping to find a model, and this might be simplistic, but we were hoping to find a model where the repair processes had been slowed down. So BRCA1 and BRCA2 are actually two distinct genes that encode for two DNA repair proteins. But these two proteins actually act to repair DNA along the same pathway, which involves homologous recombination. So most cells will rely on this pathway of repair for efficient and reliable repair of DNA double-strand breaks. And in most cases, the cells will survive this process. It's a normal process for DNA replication, as well as multiple other processes during the cell, inside the cell. But what happens when this goes awry is that the cells will actually detect this damage, and this will lead to cell cycle arrest and, in some cases, apoptosis. When these pathways are defective, the cell will then rely on another mode of DNA repair. And in that case, it's somewhat less effective and less reliable as well. And as a consequence, there can be accumulation of chromosomal defects that can lead to tumor formation. So we originally hypothesized that within these patient samples, obesity may in fact be able or we might be able to detect double-strand breaks in the DNA in relation to obesity. But as we started studying this, we realized that this is a very important population from a clinical management point of view. So these women have a very high risk of developing both breast and ovarian cancer. But as Dr. Mucci mentioned, their risk is not 100%. So there's got to be something there that is potentially modifying their risk. It might be genetic, it might be environmental, or it might be related to lifestyle. So we started thinking, could there be something that we could do to help inform the management of this disease or the prevention of disease in these women who currently have very limited options for risk reduction beyond surgical interventions, including prophylactic removal of their breast tissue and ovaries and fallopian tubes. That being said, we did look back on the literature, and there are so many conflicting epidemiological studies in this space, and I'm happy to hear anyone's thoughts with regards to this. When we first started this work, we were heavily reliant on Mary Claire King's work from 2003, showing that maintaining a healthy weight in early adulthood was actually associated with a decreased penetrance of both breast and ovarian cancer. But as time went by, we realized that there are a number of studies showing both associations between diet, lifestyle, weight, and cancer penetrance in these women. But there are also some studies showing no relationship or protective effect, other studies showing mixed results. So at the moment, it's very difficult to say with certainty that lifestyle, obesity, and other factors are able to modify risk in this population. Of course, there are a number of things that we can discuss here, like self-reported BMI, using a genetic score to predict BMI in these populations, as well as the majority of women being diagnosed with a BRCA mutation when they present with a cancer, and as a consequence, this affecting the study population. But at the moment, we don't know for sure. And if we do know anything, we certainly don't know the mechanisms behind the effects of obesity and cancer development. So in order to start tackling this question, we decided to collaborate with Dr. Iyengar, who's here today, which has been a wonderful collaboration over the years. And we were able to access breast tissue from the normal areas of the breast of BRCA mutation carriers who are undergoing mastectomy for either the treatment or the prevention of breast cancer at Memorial Sloan Kettering. What we did is we generated tissue microarrays. Today I'm presenting data for 69 of those cases. And here you see an example of what one of those tissue microarrays looks like. If you've not seen these before, the advantage of these is you can place a core of tissue for each one of your patients on the same slide. So it decreases variability. It allows you to screen many more patients at the same time, and as a consequence allows us, to be able to study biological endpoints within a larger population. So it's difficult for me to see the numbers, which means it's probably difficult for you to see as well. But basically we had women who were considered of a healthy weight or who had a BMI between 18.5 and 25, as well as women who had a BMI of 25 and above, considered to have overweight or obesity. And we had a number of clinical parameters that we are also able to assess here. And we also performed the analysis of systemic biomarkers in these patients. And the idea was that we could measure the amount of damage within this tissue and then relate it to some of these factors to generate a hypothesis as to whether obesity was able to drive cancer in these patients, and if so, which factors might be involved. Okay. So in order to be able to measure DNA damage, we really leveraged what had been done previously in the context of studies looking at DNA damage more generally, in the context of tissue irradiation or DNA damaging chemotherapeutic agents, for example, where it's routinely studied by assessing the level of gamma H2AX signal. So gamma H2AX is basically the phosphorylation of the histone H2AX. And this occurs at sites of double-strand breaks. So when the double-strand break is detected, the protein ATM will then phosphorylate the histones along about 100 kilobases on either side of these double-strand breaks, allowing us to be able to visualize actually every single break that we're seeing. So the way that we see this, and this is, oops. We can't really do that. Oh, we can. Okay, so this is an example of an H&E of one of the cores. We clearly see the breast glands, and they're surrounded by fibrous tissue. You can see a bit of adipose tissue here. And when we perform immunofluorescence, the nuclei become these blue circles, and the areas of DNA damage come up as these single foci that you see as red dots, which might be difficult to see here. And we're able to assess or measure these in an unbiased way using software. And this was a technique that was developed in our lab by Hiba Zahid when my lab was still in Melbourne. She's now a faculty member at Taiwan University. And the rest of the work that I'm showing today is the work of Priya Bardwaj, who just recently defended her thesis. So the first thing that we did is we looked at the number of these double-strand breaks in relation to body mass index of the women from whom we had collected the tissue. And here you see a very strong positive correlation between the amount of DNA damage and body mass index, suggesting that there's something going on with regards to women with obesity leading to the accumulation of these double-strand breaks. We then compared the amount of damage relative to a number of systemic factors as well as measurements that we had done. We saw a positive correlation with insulin levels as well as HOMA-2IR score, which is a measurement of insulin resistance. Interestingly, we saw no association with either local or systemic markers of inflammation, including CLS, IL-6, or C-reactive protein. But what we did see was an inverse association with sex hormone binding globulin, and this protein in particular will decrease the bioavailability of sex hormones, including estradiol. So what are the possible drivers of breast epithelial DNA damage in the context of obesity? Well, we had demonstrated that there was a relationship with insulin, and this was suggesting to us that BMI itself might not be the best predictor of damage in the breast, and something like metabolic dysfunction might be more important for the development of these breaks. So we had to look at the effects of insulin on the amount of DNA damage using two model systems, MCF10A cells, which are a normal epithelial cell. In this case, that was heterozygous for either BRCA1 or BRCA2, as well as primary epithelial cells that we had obtained from patient samples. And here you see a significant increase in the number of double-strand breaks in response to insulin levels, which are similar to what we see in hyperinsulinemic patients. But we were also interested in seeing what the local breast microenvironment, what its role might be in driving these double-strand breaks. So in order to start asking that question, we developed a 3D in vitro model whereby we were able to culture primary breast epithelial cells from BRCA mutation carriers. And they're this is the image here that I'm trying to show, and you see here in the bottom right quadrant, there are these circles, and these are really clusters of primary epithelial cells. And what we did is we overlaid these with breast adipose tissue from women who were either of a healthy weight or with obesity, and then we looked at the amount of damage within the epithelium. And you can see very clearly here that overlaying these cells with adipose tissue from a woman with obesity led to a significant increase in the amount of DNA damage in the epithelium. So suggesting that the adipose tissue is a contributor to some of the effects that we're seeing. To further characterize that, we also generated conditioned media from breast tissue from women who had undergone reduction mammoplasty. And then we treated breast epithelial cells that had a heterozygous mutation either in BRCA1 or BRCA2 with this conditioned media. And what you see here is every single dot here is conditioned media from a different patient. And we see that women who have a higher BMI, so the conditioned media that's generated from these women actually causes more damage than women who have a healthy weight. Again, supporting that idea that the local breast microenvironment might be driving some of these effects. So to start figuring out which of these factors might be important, we performed RNA sequencing on 152 cases of women who had a BRCA mutation. This is pieces of breast tissue, so we'll include all of the different cell types of the breast. And I show this image not for you to try to read any information, but really to see how the cases are clustering. So we see that in the top part, the majority of the cases are in that blue color, and these are the women with a healthy weight. Whereas in the bottom part of of this image, it's mainly the women with obesity. But interspersed within these is, cases from a lower BMI or a higher BMI. And this really, again, reemphasizes the fact that, we can't necessarily categorize women based on their BMI, especially if we're talking about metabolic health. So once we started doing the analysis of this tissue, we decided to first look at the differences in gene expression based on whether or not the cases had high or low DNA damage in the epithelium. And what we found was that a number of canonical pathways were altered in relation to having more DNA damage. And the one that really struck a chord with us was pathways related to estrogen biosynthesis. When we looked a little bit more closely at the genes involved in estrogen biosynthesis, most of us will usually look at aromatase as a surrogate of estrogen biosynthesis, but really aromatase is involved in catalyzing the last step in estrogen biosynthesis. It converts androgens into estrogens, but the breast itself doesn't necessarily have a huge source of androgen laying around. So in postmenopausal women, for example, DHEAS is the predominant circulating steroid. And these data actually show that the breast tissue has all of the metabolic machinery in order to be able to convert DHEAS all the way to estradiol, which is an interesting way to look at this, because all of a sudden the breast is its own, source of estrogen. So all of this was very interesting to us because we knew that there was a potential role of estrogen in driving the development of cancer in women with a BRCA mutation. To start asking the question of the impact of the breast microenvironment on breast epithelium a little bit differently, we also isolated breast organoids from BRCA mutation carriers. So these are only the epithelial cells. And here you see the process that we undergo to get a pure population. Basically, we take breast tissue from women, we digest away the stromal and the adipocyte component, and here you see that the normal breast organoids maintain their structure with both basal and luminal cells present. And we performed RNA sequencing on organoids from both women with obesity and women of a healthy weight. And what we found was that the changes in gene expression, or the differences in gene expression that we were seeing, were consistent with the effects of estradiol, once again, but also insulin. So it was really feeding into this idea that the breast epithelium was actually responsive to its microenvironment and factors that are elevated in obesity. What was also interesting was that Ingenuity pathway analysis identified hydrogen peroxide as an upstream regulator of this pathway, and we know that this is a DNA damaging agent, so consistent with the findings that we had made in the clinical samples. So I mentioned before that we do have some indications that estrogens might be important in driving cancer in BRCA mutation carriers. These are just two studies that highlight that. Basically, we know that women who undergo salpingo-oophorectomy, or removal of their fallopian tubes and ovaries, have a very much decreased risk of developing breast cancer. We also know that women who have developed a cancer and are treated with tamoxifen are less likely to develop a secondary tumor while on their treatment, suggesting that tamoxifen use, even in BRCA1 mutation carriers who have been generalized as developing only triple-negative cancer, which is not true, are able to benefit from reductions in estrogen receptor signaling. So to start asking this question a little bit differently, we looked at breast tissue. Again, this is normal breast tissue from BRCA mutation carriers. And the first thing we wanted to do is really see whether the estrogen receptor was there. So we know that it's a normal component of the breast. It's really what drives many of the changes in mammary gland development at the time of lactation. It has many, many roles in the normal biology of the breast. And we weren't surprised to see that the estrogen receptor was expressed in the mammary gland of both BRCA1 and BRCA2 mutation carriers. We were also interested to see that the majority of the DNA damage foci that we were seen now in green, actually tended to co-localize in cells that had increased or expression of the estrogen receptor seen in red. So we went back to our organoid model, and when we treated these with estradiol, we saw a significant increase in the amount of damage within these cells. And interestingly enough, this was reversible with the selective estrogen receptor degrader fulvestrant. Now, this is all well and good. You know, it's really pushing or emphasizing a potential role of estradiol in causing DNA damage in these patients. But, you know, the options for prevention in this space are relatively limited. You would never give something like fulvestrant in a prevention setting due to the side effects associated with its use. So we started to be a little bit more creative. Of course, you know, reducing weight and improving, you know, lifestyle in general is probably the most desirable approach to all of this, but it's not necessarily a practical approach in a patient population. We had previously demonstrated that metformin, the anti-diabetic drug, was able to suppress aromatase expression in the breast. So we decided to start looking at the interaction between aromatase and the epithelium and to see whether metformin itself might be useful in this setting. Now, this graph here is just showing that there is a relationship between the number of double-strand breaks and aromatase transcript expression in breast tissue of BRCA mutation carriers. And if we use these tissue explants, which contain the microenvironment in addition to the epithelium, and we suppress estrogen receptor activity, we continue to suppress DNA damage. So we believe that, you know, taking into account the microenvironment in this setting might be very important. So we then went on to test the effects of metformin, and this is at micromolar doses, which are pharmacologically achievable. And what we found was that metformin was able to suppress in a dose-dependent manner the formation of these gamma H2AX foci in the tissue explants. So these are the explants that have the microenvironment, but had a much less dramatic effect in the isolated organoids, suggesting that the effects are really indirect via the microenvironment. And because of what I mentioned before, we also looked at the levels of steroids within these explants. We measured estradiol and testosterone levels as well as other steroids. And we saw a dramatic decrease in estradiol within the tissue with the treatment with metformin. And obviously this has implications beyond, you know, what we're studying here in the context of DNA damage in this population. And we were seeing an increase in testosterone, which would be consistent with inhibition of aromatase within the breast tissue. So we then turned our attention to a mouse model because we were hoping to be able to start act on some of these findings in, with new interventions. So we developed a BRCA1 heterozygous mouse on a C57 black 6 that's more likely to develop obesity, and put these mice on either a low-fat diet or a high-fat diet. And after 22 weeks, the mice gained more weight. Keeping in mind that these are ovary intact mice because we had an interest in what was going on in the fallopian tube and the ovaries as well. But despite being ovary intact, these mice still developed a certain degree of glucose intolerance, and they had accumulation of mammary fat. Gene expression within the mammary fat pads with high-fat diet was also very similar to what we see in women with obesity relative to those that have a healthy weight. So we then went on to look at DNA damage in the mammary gland of these mice and saw a significantly higher level of damage in the high-fat fed mice relative to the low-fat fed mice. And this more closely correlated with mammary fat pad weight than it did with overall weight, again emphasizing that measures of height and weight might not be the best way to assess adiposity and metabolic function. So far I've really shown you that a number of factors associated with obesity can cause damage to the DNA of cells that are heterozygous for BRCA mutations. But we really haven't pushed it that far to say, well, this is actually going to lead to cancer. We're not quite there yet, but we did utilize this model to start asking that question. What we did is we put these mice on a low-fat or a high-fat diet, and then we introduced an medroxyprogestin pellet, which will prime the mammary gland for tumorigenesis, and then provided these mice with an altered regimen of a carcinogen that's previously been shown to cause mammary tumors. Now, what we did is we reduced the amount of the carcinogen so that we might still be able to see modification based on the diet itself. What we found was that the mice on a high-fat diet, and these are mice with a BRCA heterozygous mutation, had increased penetrance relative to the mice on a low-fat diet, with, I'm struggling to see the number, 86% of the mice on a high-fat diet developing tumors versus 60-something% of the low-fat diet ones. In addition to that, we were also seeing that the mice on high-fat diet were developing these tumors sooner. So there was a decreased latency in relation to the development of the tumors. And what was interesting, actually, is once the tumors developed, they actually grew at the same rate, no matter whether they were from a low-fat diet mouse or a high-fat diet mouse. So this was suggesting that the effects we were seeing here were not necessarily due to the tumors in a high-fat setting growing faster. It might actually have something to do with how quickly they were developing. So this is the last part of my talk, and it's a very small snippet, but we were wondering how broad the effects were of obesity in BRCA mutation carriers. So we also decided to look at DNA damage in the ovary and the fallopian tube of BRCA mutation carriers. So these were tissues that were obtained from prophylactic surgeries, so women who did not have cancer. And what we did is we assessed DNA damage either on the surface epithelium of the ovary or the fimbrial region of the fallopian tube. And the reason that we also assessed the fallopian tube is that there's more and more evidence that ovarian cancers actually don't originate from the ovary itself, that they're originating from the fallopian tube. And I think this is what's justifying removal of the fallopian tube only as a preventative for the development of ovarian cancer in this population. What we found was that having overweight or obese was actually associated with a significantly higher level of DNA damage in the fallopian tube relative to being of a healthy weight, and that was not the case in the ovary. And I think that really supports that idea that changes, or the fallopian tube might be more susceptible to changes that lead to cancer formation. So with that, I'd like to, introduce or reinforce this idea that in thinking about cancer treatment or cancer prevention, it's important to start thinking about ways to be targeting this in a more holistic way. So for a long time, we were treating the tumor cells. More recently, we've become really interested in immunotherapy and involving the microenvironment. And if we think about endocrine therapy, it was already doing that. But in combination with something that would work at a whole body level, we might be more effective at either reducing the risk or better treating the disease. And there are some approaches that would allow us to do all of these things at the same time. So with that, I'd like to acknowledge the lab. I'd like to reiterate that Dr. Bardwaj did most of this work, and she's an exceptional scientist. I know she'll do great things. I'd also like to acknowledge Neil again, because this collaboration would not be possible without you, as well as the funding agencies, the clinical teams, both at Cornell and at Memorial Sloan Kettering, and the patients who donated their tissue, as well as Michael Polak, who did the biomarker measurements, and Manho Choi, who did the steroid measurements for us. Thank you. I'm happy to take any questions.
Thanks, Kristy. I really love this body of work that you've generated. So a question about metformin, and I know you didn't have too much time to get into this, but what do you think the driving mechanism is for the protection against the DNA damage that you observed in your models? And is ER alpha expression required for metformin to be protective in this setting?
Yeah, those are excellent questions. I think there's been a lot of debate as to, you know, the potential effectiveness of metformin in many settings in the context of breast cancer. So in the context of the work that I presented, which is purely in an explant model, so this is ex vivo, so we have to so insulin will not be modified by metformin in this system because it's not systemically administered in this context. So our original rationale for doing this was to see whether metformin via the suppression of aromatase in the microenvironment might lead to a decrease in DNA damage, as we had shown that estradiol was able to cause this damage. But the more that we're doing this work, the more we realize that metformin has the potential to act at multiple levels. There was a really nice study from Michael Pollack, maybe about 10 years ago, showing that metformin was able to reduce the amount of damage in a normal epithelial cell, not a BRCA heterozygous cell. And it did this by reducing the amount of reactive oxygen species within the cell. So in our organoid models, we see some reduction, so that would be epithelial specific, but it's not as dramatic as what we see when the microenvironment is present. So I think that there's probably a combination of effects of metformin in the microenvironment and the epithelium. And then, you know, the reason I get excited about it is because, you know, if you think about whole body treatment with metformin, you would also expect to reduce hyperinsulinemia in patients who are either pre-diabetic or, you know, not currently being treated. And as a consequence, you might really be targeting, you know, this process from multiple different angles.
Thank you.
Hi, how are you? Very nice talk. I wanted to hear your comments about about the animal model when you treated with obesity that they developed cancer, but then like the growth was not enhanced. Do you have any hypothesis of why? Because I don't know, there has been many discussions on like how obesity could fuel or the pro-inflammatory or the insulin or others.
Yeah, we're still figuring that out. And it's actually interesting because there is a huge immune infiltration in the tumors in relation to obesity or to high-fat feeding. The one thing that I have to say potentially as a disclaimer is that the location of the tumors tends to be different in the low-fat mice versus the high-fat fed mice. The low-fat fed mice, although it's not absolute, they tended to develop tumors more in the thoracic area, whereas the high-fat fed mice tended to develop more in the inguinal area. And there have been studies suggesting that there might be, you know, a different crosstalk for these ones. So we've also looked at hormone receptor status in the tumors. And there does seem to be a reduction in hormone receptor positive tumor formation in the high-fat fed mice, which might be more consistent with some of the data that has come out showing that obesity influences triple-negative development, especially in younger women. But I think that's really where we're at the moment with regards to our understanding of how these tumors develop. These are also BRCA1 heterozygous mice. And, you know, it's interesting because on low-fat diet, the proportion of triple-negative tumors is actually quite similar to the human population. So there is a proportion of triple-negative, but there's also a proportion of hormone receptor positive that allows us to kind of start looking at these different tumors using this mouse model.
Okay. Thank you. Thanks very much.
I'm going to be sharing a question from the online community. Women with polycystic ovarian syndrome have overlap with higher rates of obesity, insulin resistance, and hormone imbalance. They also have a higher risk of breast and endometrial cancers. Do you think this might be connected in some way, suggesting that they may be a patient population that would benefit from BRCA testing, even if they don't have a first-generation relative with cancer?
That's a very interesting question. And it's also interesting to see how this field has progressed, because when we started studying the obesity-cancer relationship, it was very difficult to say anything about cancer risk in the PCOS population. Now there's a lot more evidence. And as you've suggested, there are a lot of risk factors that are common between them. But to my knowledge, I don't know that there is any established link between carrying a BRCA mutation and development of PCOS. But maybe it's not been tested. So that's a very interesting point. All right. So I think I'm going to wrap us up for now. We have a short break, and I think we're supposed to reconvene at, is it 4 o'clock? Does that make sense? Yeah. So we'll have a short break and then come back for the second part of this session.
Great. So it seems, unless I've correct me if I'm wrong, that a real common theme between all the speakers in this session has been the importance of DNA damage in cancer development, because it can contribute to genetic instability, which is a hallmark of cancer. And so what I get from that is then it's important to try and reduce DNA damage in normal tissue through things like weight management, coffee consumption, perhaps. So my question is there anything we can do if there is already some DNA damage? So say if inflammation drives DNA damage, that's not then causing cancer yet. There's sort of a pathway that it then creates genetic instability and extra mutations favoring cancer. But is there something we can do to repair that DNA damage if it's there already?
So from a biological perspective, we've been thinking about this. I think that the damage will get repaired one way or another. If it doesn't, the cell will be targeted for cell death. So the question is, how well is it being repaired? And are any of these factors influencing the mechanism of repair and the quality of repair? And can you start acting on those processes specifically? So I think there are multiple ways of looking at this. Can you reduce the amount of damage to begin with? And can you improve repair mechanisms? And I don't think we know the answer just yet. I think it's, obviously a $1 million question. But I agree with you. It's, at the basis of a lot of, what we would expect leads to tumor formation. Does anyone else? Actually, along those lines, what's interesting and was highlighted in our discussion today is that, most of our hereditary cancers are, or many of them, are a result of mutations in these DNA repair pathways. So one of the areas that I thought we could talk about as well is what's the impact of these heterozygous mutations that may or may not lead to haploinsufficiency in other cells that don't necessarily lead to cancer. So do we know about the impact of these mutations on the immune system, for example, on response to exercise, on the production of prostaglandin E2, on any of these pathways that could also help explain why these tumors are exacerbated in these patients?
Eduardo, do you have a comment on that? Because you're doing a lot of mechanistic work in that space.
Yeah, I can try, but I don't know if there is much information, right? I think we have learned a lot about what it means to have a complete deficiency in these DNA repair systems. But as Dr. Brown was mentioning, haploinsufficiency is the big elephant in the room, right? So I think there is relatively small amount of information on what are the consequences on doing it, on happening that, right? And there is somehow conflicting data. In my field, which is basically mismatch repair deficiency, for example, there are data published recently by the Sanger, where they were looking at degree or genomic instability in general in colonic mucosa of Lynch syndrome carriers, and they didn't see an increased mutational burden. But that is at the epithelial level. I think is, for example, fascinating the comment of whether or not this could influence other tissues that may have higher rates of proliferation, like in the immune cells. We published in Cancer Research a year ago using the Lynch syndrome mouse model. What was the gene expression profiling of haploinsufficient colonic stem cells, and we clearly saw that these cells are fundamentally, or had a transcriptomic profile that is fundamentally different to the normal cells. So that makes me feel that there are obviously consequences to having only one functional copy of the gene, but I think that the data is still there at the level that we can definitely solve that question. I hope that more information coming from the Human Tumor Atlas, the Pre-Cancer Atlas that is basically doing single-cell genomics will help understanding that better. But I totally agree. I think that it will be very interesting to see what is the differential effect across the different tissue types. And obviously what will be the influence of other factors on those.
I just wanted to add, I mean, it's a little bit different than the question that was asked, but we've done some work looking at gene expression profiling in prostate cancer tumors, but also in the adjacent normal tissue and in the stroma as well. And for example, for physical activity, we actually see a much stronger signal, not in the prostate tumor, but rather in the adjacent normal tissue, which might suggest sort of kind of along the lines that these types of benefits of diet or healthy weight or physical activity may have much of effect on the microenvironment in which the tumor is living, as well as the tumor itself. So I think there's some really interesting areas there. I mean, that was sort of an epidemiology study. It would be interesting to see how that recapitulates, say, in a animal model, for example. But I think that's interesting. The other thing I was going to say was that a colleague of mine has done work taking serum from individuals who are on, for example, short-term intervention studies, and then taking the serum and exposing prostate tumor cells to that serum and it actually was interesting to see that those tumors grew more slowly than those from the same bloods from the same individuals prior to the intervention. So again, I think there are some interesting different approaches that we need to think about, and I do think it's going to take sort of multidisciplinary teams to kind of really validate some of the findings and bring the research forward.
I was going to ask actually about risk reduction and markers for effective risk reduction. So in the absence of following a patient to, disease occurrence and progression, what do we have as a trustworthy biomarker of risk, but also risk reduction? And if we don't have any of those, what do we need to be looking at? Do we need to be looking more closely at the tissues of origin of cancer? Or can we rely on systemic markers that are less invasive, but potentially less specific?
Yeah, I'm happy to jump in. I mean, it's hard. I mean, even prostate cancer, we have the PSA blood test, and while there was some controversy in the past, it's known via randomized data to save lives, and there are certainly limitations to it, but that's essentially what the studies have shown. It hasn't even shown that if you do some kind of intervention, be it behavioral or whatever it is, to lower a PSA, that you then affect risk. It's simply that, who knows how much money was spent, enormous trials were done with over 20 years of follow-up to show a mortality benefit. So in my mind, we don't have it yet, but it's all going to relate back to mechanism. It has to relate to the tumor itself and substratifying patients, and maybe something like a screen-based biomarker like PSA will end up being important, but it'll have to be within the context of what that individual patient has in terms of, be it the epithelium at risk or maybe a small kind of pre-malignant or low-grade tumor that's already kind of occurred.
The panel has disappeared from our screens, but if you're there, there you go. And did you have any comments with regards to that?
No, I fully agree. I think it is unfortunately somewhat challenging to think about intermediate endpoints. Just thinking about prostate cancer as an example. Also, from the time someone gets diagnosed to prostate cancer, there really needs to be, say, 10 to 20 years of follow-up to see what's going to happen to them, particularly if they're diagnosed with non-metastatic cancer. And so it would be a value, it seems, to have some sort of intermediate marker so that you could sort of either do a clinical trial or even in our epidemiology studies have a good surrogate of disease progression. But I don't think we're there quite yet, it seems like.
Yeah, I totally agree with that. That's one of the things that we have been pushing in all our clinical trials, trying to interrogate what happens in the normal tissues, either normal mucosa, normal blood, trying to associate any effects on the intervention that we are looking at with those biomarkers, with the hope that when we design a phase 2, we don't have to rely on 10 years of follow-up to come up with that conclusion, right? I totally agree that I don't think unfortunately, we're not close to it.
Nigel?
I was just going to say that the other thing I've noticed, we're kind of talking about themes and different talks, and themes that we see in cancer, nutrition, physical activity interventions is inflammation, right? So we know that there's some effect of DNA damage in terms of disruptions in the tumor and how that leads to inflammation. We talked about disruptions in the immune system and how that leads to inflammation. Of course, if we ramp the immune system too much in cancer treatment, that leads to inflammation. And then, of course, disruptions in cancer metabolism through obesity, through different dietary habits, that leads to inflammation. So really understanding that, to me, besides DNA damage, seems to be very important and very common theme. Although I was just talking at lunch to one of the colleagues, we always have to decide what inflammatory markers we want to measure and how deep do we need to go into all the things that affect that to be able to say, this is our intermediate marker. And of course, the microbiome is involved in inflammation. So we have a set of tools, but one of the challenges for people in epidemiology and analysis and trial design is how to integrate these things together. And that's another theme I kind of noticed from the talk. Well, why don't you do physical activity with diet? Or why don't you do coffee with this? And why don't you do that? But the problem is that we have to look at some of these things in isolation to get some of those signals to understand.
Nigel, you've been at the mic for a little while. Would you like to join us?
No, I'm good. I guess I just want to remind everyone that how far we've come in a very short period. So when we released the third expert report in 2018, and we had lots of internal discussions about the messaging of the report, and at one point there was a sort of assumption that was, going to be part of the message, that this is particularly important for people at elevated risk through family history. And I was like, hang on, we don't actually have that evidence. And intuitively, people are like, well, it must be. But then on the flip side, the influence of family history and genetics could actually overwhelm it. So the fact that you are, this panel has done this work, and that was 2018 we were doing that, and we've had a pandemic in the middle and all the rest of it. So, I mean, we have come a long way. And I guess my question is, having sort of got this preliminary data, where are the big opportunities? What is it we need to be doing? And that's just a fairly open-ended opportunity to write your next grant.
I just want to kind of add to that question, and where are we going? Because it seems to be that, and the clinicians in the room can tell me if I'm wrong, but it seems to be, with regards to clinical care, that there's less and less of an emphasis on extreme surgeries, So it's about, reducing the amount of damage. We know that, for example, women who carry a BRCA mutation don't necessarily gain a survival advantage from having prophylactic mastectomy. But does this mean that you know, we need to be working even harder right now to make sure that we have these adjunct, you know, risk reduction strategies, so that when, you know, these women start potentially having a slightly higher risk of developing cancer because they're not being, you know, the tissue isn't being surgically removed, that we can at least, you know, influence that to some degree.
Sorry, man.
Yeah, I'm happy to comment. I agree. I think at least in urology, we're constantly looking to limit side effects, but keeping in mind that cancer mortality is the primary goal. And once you've kind of met that cancer mortality objective, you know, then you're looking at, okay, how's this patient going to live? How's everything going to go? So I would say we are keeping cancer mortality sort of in our line of sight. But the idea of, you know, repeating this again, just substratifying, like what Dr. Mucci's done with genetic risk and showing, you know, parsing out which patients may benefit from certain interventions where then they don't need a large surgery or something of that nature, I think is critical. And I think the other big question is, a lot of what you're looking at is just fascinating, of how does the cancer develop? And then is there a difference once the cancer exists and to how it progresses and potentially causes a problem? Because that may be something that, you know, it's literally different potential interventions or behavior of, you know, the normal versus the tumor epithelium, and then how you actually impact that. So I think that further kind of stratification is what we need, and it's, you know, based on your work of mechanism.
Yeah, I think the biggest challenge to get back also to Nigel's point is, like, what are the next steps? So we have to be able to clearly define what the interventions that will be beneficial are, you know, for which population. What are the biomarkers that will allow us to assess risk reduction? How can we then modify, you know, factors that we can't modify, you know, by lifestyle? You know, I'm constantly thinking. So one of the asides of our work is that we actually discovered that in a heterozygous cell, so a BRCA1 heterozygous cell, the influence of obesity, or the impact of obesity, is actually to decrease gene expression of about 50 different genes that are involved in DNA repair. So it's like obesity is actually pushing the cell towards, you know, something that mimics loss of heterozygosity, at least at a gene expression level. We're trying to figure out whether that's true at a functional level. But, you know, I feel like sometimes we're missing it if we're not studying the population that actually has obesity or something like that. We might be missing those intricate details. But from our perspective is, how can we then go back in and restore that, you know, capacity for repair if it's defective?
I'll just say that I think the other theme that I've seen that I'm always very excited about, especially is important for a cancer audience or a chronic disease audience, a cancer patient audience, is that a lot of the things that we're studying and we're interested in understanding can work synergistically with other pharmacologic treatments that we know are effective. And so I'm also very intrigued by how we can continue to study that, because I don't necessarily think that a lot of us are out here trying to replace effective drugs with diet, though that often is the way the message sometimes is delivered in the press. But I think that, you know, Dr. Cantley's work and what Eduardo is working on and really thinking about, of course, Eduardo wins the naproxen exercise trial, but anyway, thinking about these things, I think that's another exciting area for high-risk patients where that could be really effective.
I'm not going to come up there, but I'm going to quite happily sit in the side. The other thing, Kristy, you were very sort of cautious about saying we're not saying this is definitive yet. But when you look at it, we've got, you know, very suggestive evidence in breast cancer. We've got good evidence in colorectal cancer. We've got good evidence in prostate cancer. That's 3 of the big 4. And smoking is so confounded by, lung is so confounded by smoking. So I really think we are getting to the point where we may even be able to make specific, strong recommendations to people that do have that family history, and certainly to get away from that sort of fatalistic, well, it's my genes, there's nothing I can do. Yeah. I think basically bravo for the work you're all doing.
So, yeah, I'm not sure how we're doing for time, but I think, this was an interesting discussion, and I'm sure we can continue it after this panel. I'd like to thank everyone. This is the last session of the day, so there will be poster presentations again tonight. I'm not too sure what the time is, but...
And we're transitioning straight from here to the reception outside. It kind of happened organically yesterday, so rather than sort of having that defined break, people can just go out and start enjoying the entertainment.
Thank you.
Thank you so much.
Thank you.